Hello everyone. Today we are going to discuss the insertion of DNA and ligation using blunt ends, cohesive ends. This insertion of DNA into the what that is a host organism is the important step, but the joining the two fragments, that is the what the foreign DNA which is spliced, and the what the DNA of the vector molecule. This should be ligated and or joined. And this ligation efficiency, it depends on the DNA ends in the reaction. Means by which enzyme or by which restriction endonuclease we are cutting the enzyme. Either you will get the sticky ended molecule with unpaired nucleotides, or you will get the blunt ended molecules with what that is a paired nucleotides. So if the unpaired ends are there, that we called it as a sticky ended molecules. And if the complementary sticky ends are there, then ligation is very efficient. Why? Because they will identify the complementary sequences, and annealing of the complementary overhangs brings the 5' phosphate and 3' hydroxyl into the close proximity. So annealing between the what that is the complementary sequences or the complementary overhangs. And so that the ligation will be very easy in terms of in case of the what that is a sticky ended molecules. While the other type, if the both the molecules are blunt ended, that is both the DNAs, that is the DNA of the foreign, that is the DNA of an interest, and the other one DNA of the vector. If both are the what the blunt ended, then ligation is inefficient, and it needs high concentration of enzyme ligase and also high number of the what that is the DNA or the high concentration of DNA. And molecular crowding reagents like polyethylene glycol improve the intermolecular ligation, then dilute to promote the intramolecular ligation. So now what are the techniques which are useful for joining the DNA molecules together? The first one is what that is the ligation of the blunt end, that we have already discussed. The ligation of the sticky end is very efficient, and annealing occurs within the what that is a complementary overhangs which brings 5' phosphate and 3' OH into the what close proximity. Now we'll discuss the what the technique or the ligation of technique of the what that is a blunt end. The ligation reaction can take place between the blunt ends of two DNA fragments between complementary and overlapping sticky ends. In this figure, if you observe the what if this DNA is cut with the molecule that is what HIND, then what HIND1, this what will happen? This is a recognition site, and you are getting the no sorry HIND2, and we are getting the what that is the two DNA fragments which are blunt ended molecules. These are the paired nucleotides. These are the blunt ended molecules. To join such molecules, what you have to do? The some restriction enzyme like HIND2 cut the DNA in the middle of recognition sequence, leaving the blunt-ended DNA fragments. And these fragments had show no tendency to stick together. The ligation of blunt end is possible in test tube, but reaction is not very efficient. Since T4 DNA ligase from E. coli cannot bring the two DNA molecules together, only a chance of association can bring the two ends together. Therefore, blunt end ligation has to be carried out at high DNA concentrations to increase the chances of two blunt ends contacting each other. So therefore, we have to choose the what another process for ligation. At low DNA concentration, there is a tendency for each fragment to form a circle with itself, and high DNA concentration favors the recombination between foreign DNA and the plasmid DNA. One advantage of blunt end ligation is that DNA sequences are joined without introduction of any DNA between them. And the disadvantage is that which pairs of blunt ends becomes joined cannot be controlled. That is by the only it is only by chance they will get attached to each other. Then this is a common procedure that we use in the recombinant DNA technology. This is plasmic vector. This is the cleavage site where you have cut, and this is the foreign DNA, and these are the unpaired bases, and that we call it as a sticky ended molecule. If that sticky ended molecule, there is what this this sticky ended molecule, what that is the complementary sequences will anneal together, and it will form the what that is the seal the nix. And so that such type of the what that is a ligation, that is a what the ligation with the what the sticky ended molecule, it is very much easier. But in case of the blunt ended molecule, it is not very easy, and therefore we have to use the certain other molecules like the linkers and the adapter molecules. What are these linkers? Linkers are the what? The short synthesized sequences of double stranded DNA. And typical linkers, what are these? These are the synthesized sequences of double stranded DNA. And these typical linkers are also the blunt ended molecule, but contain these linkers having the what? This site, that is the what? BAMH1 recognition site. This is the BAMH1 recognition site, and that can produce this when this this molecule is subjected to action of the what that is a BAMH1 enzyme. What will happen? It will make the what? It produce the sticky end molecule. The blunt-ended linkers are first ligated by the DNA ligase to these ends of the blunt-ended molecule. This is the blunt-ended molecule. These are the linkers. These linkers are attached to the what that is a BAM. These linkers are attached to the what that is a blunt-ended DNA molecule, and this entire molecule is subjected to action of the enzyme that is a BAMH1. Why? Because this BAMH1 is such an enzyme which such a restriction endonuclease which form the sticky ended molecule. So that the reaction involves a blunt ended ligation. It can be carried out efficiently because it is possible to add high concentration of synthetic linkers to the ligation mixture. More than one linkers may become attached to each end of DNA molecule, and the action of the what or the digestion of the BAMH1 cleaves the chain at the recognition sequences, and thus this results in the DNA fragments with BAMH1 sticky ends with large number of the cleaved linkers. These are the large number of the cleaved linkers, and this is the sticky ended molecule. By this method you can use the word that is a you can convert this word blunt ended molecule into the word that is a the sticky ended molecule by using the linker. What are these linkers? These are the blunt ended, that is the short synthesized sequences of the double stranded DNA, and these linkers specifically having the word the restriction endonuclease site for the word that is a BAMH1, and this BAMH1 it cleaves the DNA in a such a way that it makes the way into the what that is a sticky ended molecule. Then this is the what the cloning of the foreign DNA by adding the what that is a linker. In this case, we have added the what that is this BAMH1. Again, this EcoR1 is an enzyme which also cuts the what this DNA fragment into the what that is a sticky ended molecule. So these linkers are attached to the what that is attached with each other. This is the foreign DNA and this linker molecule. Foreign DNA is a what the blunt ended. This linker molecule is blunt ended. We are adding these, and it is a final molecule which is a blunt ended. That is, with these are joined together with the help of T4 DNA ligase, and now this entire molecule is subjected to action of EcoR1. EcoR1 will produce the what the sticky ended molecule, and this sticky ended molecule now it can be ligated into the vector molecule. So in this way the blunt ended molecule is converted into the what the sticky ended molecule. So this is a way by how to use the word that is a linker molecule. Then second molecules which are used for ligation of the blunt ended molecule, these are the adapters. In double stranded DNA molecule, each strand has 5' phosphate at one end and 3' hydroxyl group at another end. The two strands are anti-parallel so that each end of double-stranded DNA molecule has one phosphate end and the 3' that is at 5' end there is a phosphate or pyrophosphate and at 3' end hydroxyl terminal is there. Ligation normally occurs between the 5' phosphate and 3' OH. The adapters like linker are the synthetic oligonucleotide. These adapters are also the synthetic oligonucleotide, but unlike linker, adapters have one blunt end and one sticky end. Means the linkers were having both the that is what the blunt end, but these linkers are having the one blunt end and the one sticky end. The sticky end of adapters would have tendency to pair with one another and ligate to produce a blunt-ended dimer. And to prevent this dimerization, 5' termini is modified to a 5' that is a phosphate is removed and instead of that the hydroxyl terminus is added. And since DNA ligase cannot form phosphodiester bond between 5' hydroxyl and 3' hydroxyl end, ligation does not occur. So these adapters are what? These are like linker, the synthetic oligonucleotide, and unlike linker, adapter have one blunt end and another sticky end. Such modified adapter molecules can be ligated to molecule by the DNA ligase but do not ligate themselves. Why? Because we have modified the what this 5' terminal end by the what hydroxyl group and the abnormal 5' OH end of the adapter molecule is now treated with enzyme polynucleotide kinase which converts again it into the natural form that is a 5' pyrophosphate form and sticky ended DNA fragment can now be inserted into appropriate vector. Here, this is a blend ended DNA molecule, and these are the adapters, which having the what that is a one sticky end molecule and the one blunt ended molecule. Now, what this ligation or the dimerization may occur, and to prevent the dimerization of these, we have changed the what that is a here. If this is a 5' end and this is a 3' end, we are what these are substituting the what this pyrophosphate or the phosphate from this 5' end with the hydroxyl group and so that hydroxyl group and hydroxyl group will not able to form the what that is a this dimerization and so that the sticky ended DNA fragment these after this is a what the blunt ended DNA molecule these are the adapters and these adapters that this is a what OH end and this is a what that is a OH chain and so that how these attach and with the help of the DNA ligase and again after that this hydroxyl group from the what that is a both the end that is a both the 5' end it is removed with the help of the what that is a or substituted by the phosphate with the help of the enzyme that is a polynucleotide kinase. So in this manner the what adapters can be utilized to make the what that is a blunt ended DNA molecule into the what that is a sticky ended DNA molecule which can be ligated or with the vector molecule very efficiently. This is the cloning of the foreign DNA by adding the what that is adapter molecule. This is a what our DNA and this is a 5' end having phosphate. This is 5' end phosphate, 3' end hydroxyl. This is the 3' end hydroxyl group and this is the or what the adapter. Adapter having the one that is a what this is a one blunt end and the another one is a what the sticky end molecule. And after addition of these, what all these when you are adding this, what will happen? This both the hydroxyl group will be attached at this portion and the what that is a here also the what that is a this molecule having the what that is a both the hydroxyl group. Now this is subjected to action of the what polynucleotide kinase and so that what will happen it will form the what that is a sticky ended molecule which can be ligated into the what that is a the plasmid DNA or the what that is a the vector DNA molecule. The advantages of this is you do not need to treat the adapter modified DNA with a restriction enzyme. So this is the what how the what adapters can be used. But instead of using the restriction enzyme, you have to use here the what that is a polynucleotide kinase to replace the what this hydroxyl group from the what that is a 5' end with the what that is a phosphate. So this is how the what the adapters are used for the what this. Then the what terminal transferase can be used to add the polynucleotide. Terminal transferase to is or the enzyme it is used to what that is add the what that is a polynucleotide tails. Here the what this is the what the blunt ended molecule. This is 5 dash and 3 dash 3 dash 5 dash. This runs in this direction. This will run in this. That is what lambda. That is what exonuclease or the activity of the exonuclease. It will bring about the what that is a few nucleotides from the what that is a these ends, and so that the what by action of the what that is a terminal transferase, what you will add here the what that is a polynucleotide tails. That is the what here T T T T here also at this 3 dash and T are added. This is the what the tail which are added due to that by the help of the what that is a terminal transferase, and after that the homopolymer tailing will be there, and annealing will be done to what that is attached. So how this terminal transferase is also used to what that is a what make the what that is a blunt ended molecule into the what that is a the what the sticky ended molecule by using the what that is a this that is a polylinkers which are at the attached at the what that is the end of the molecule at the 3 dash end with the help of the what that is a terminal transferase. This is the homopolymer tailing. Just we have discussed that is a polymer with identical subunits, that is a polydeoxyguanone, or that is a what the polythymidine. The enzyme terminal deoxynucleotidyl transferase is used to add the homopolymer tail consisting of series of identical nucleotides at 3' end. Here the at 3' end the polynucleotidyl what that is a thymidine, it is attached. Sometimes you may attach the what any type of these, that is a polydeoxyguanosine or polydeoxythymidine like the homopolymer tails. You can attach polycytosine tails commonly attached to the vector molecule, and the polyguanosine tails are attached to the insert DNA. So that the what when the two types of the DNA molecules are mixed together, base pairing takes place between the complementary homopolymer tails, that is what the cytosine to the what guanosine pairing to produce the recombinant DNA molecule. In practice, the poly-DC and the poly-DG tails are not usually of the same length. The recombinant DNA molecule therefore has the nicks as well as the discontinuities at the junction, and therefore repair takes place in two steps. In the first step, the nicks are filled by using the clinopolymerase, and in the second step, the phosphodiester bonds are synthesized in discontinuities using the what DNA ligase. So the another technique that is for the what the making the what that is or attaching the or the ligating the what the blunt ended molecule or the blunt ended DNA molecule is a what the homopolymer tailing. First is the what first method addition of the what the linkers. Then second method addition of the or the use of the what that is a adapter molecule, and the third method is the what homopolymer tailing. These are the three methods which are useful for the what the ligation of the blunt ended molecule and to the what that is a vector, or converting the what that is a blunt ended molecule into the what sticky ended molecule, and then ligating it very efficiently. Then next is the what the how to after the what that is a when you are ligating these two molecule, you got the what the recombinant molecule, that is what you have added the what the these two molecules, or you have joined these two molecules in this way, and this final molecule which having the what the foreign DNA and the what that is a DNA of the vector or the vehicle molecule or the carrier molecule, that we called it as a recombinant DNA molecule. And now this recombinant DNA molecule you have to transport into the what or the introduce into the host cell. Generally the host cell it is used is a what that is a bacterial cell. And in bacterial cell, if you are transferring the what that is recombinant DNA, the process it is called as a transformation. You might be knowing the Griffith experiment of the transformation of the what that is a bacterial transformation. Here the what in this process what happens that these the plasmids of these DNA is extracted from the what that is a antibiotic. It is not antibody, it is an antibiotic resistant strain of the E. coli, and the DNA consists of chromosomal DNA and the plasmid DNA. This plasmid it confers the bacterial resistance to the bacteria to the different antibiotics like the tetracycline. The chromosomal and plasmid DNA show differential binding to ethidium bromide, and the plasmid DNA fraction is separated from the chromosomal DNA, and the cell membrane of E. coli tetracycline sensitive cell is made permeable with the help of the treatment of the calcium chloride. We'll discuss the how the what that is you can introduce this the recombinant DNA molecule into the what the bacterial genome. Then this bacterial or this plasmid bound to the cell exterior, and this is a what the competent cell, which is a what the antibiotic sensitive cell. This antibiotic cell, that is what the action of the what that is subjected to the action of the what that is a calcium chloride treatment, and suddenly that is what it is rise to the 42 degrees centigrade for the 2 minutes. What will happen? It forms or makes the what this uh, plasma membrane, that is what the permeable or the cell wall, it makes it the what that is a permeable to the what this plasmid, and so the plasmid will enter here. So the what how that is done. This is a tetracycline sensitive cell, and it is subjected to action of the what that is a calcium chloride, and then suddenly the temperature is increased, and so that it will make the wall porous, and the what that is this will bring the what that is this whatever the what the plasmid, it will enter within the what cell, and it will transform the cell. What is the transformation? That transformation is defined as the process of taking up of the naked DNA from the surrounding into the host cell. That was demonstrated in the Streptococcus pneumoniae by the what that is a Frederick Griffith, and for this transformation we are using in the what recombinant DNA technology. Here we are transforming the what that is a bacterial cell by using the what that is a induced competence by action of the calcium chloride. Induced transformation is done. This calcium induced chloride induced transformation involve making the E. coli cell competent to take the plasmid DNA. Since DNA is very hydrophilic molecule, it won't normally pass to the bacterial cell membrane. In order to make the bacteria to take the plasmid, they must first be made competent to take the DNA molecule, and this involves the addition of calcium chloride to the cell suspension, which enhances the binding of plasmid DNA to the lipopolysaccharide of the what that is a bacterial cell. And positively charged calcium ion attract both negatively charged DNA and negatively charged group in the lipopolysaccharide inner core. The plasmid DNA can then be passed into the cell upon the heat shock, wherein the cells are cooled to low temperature first at 4 degrees centigrade, and then heated to high temperature at 42 degrees centigrade. Means this action, or this is it is subjected to the action of the what that is a cold treatment. That is what that is a cold treatment, that is a calcium carbonate, the sorry calcium chloride, and the what this calcium chloride, and at what temperature, for 4 degrees centigrade, and then it is suddenly heated to high temperature at the 42 degrees Celsius for near about 2 minutes or for a short time, and this causes the what that is a bacteria to take up the DNA, and then such type of the cells then plated on the what that is a the culture media, and with the appropriate antibiotic after inoculation at 37 degrees centigrade for 1 hour to enable the transport gene to express in E. coli on recovery of the cell. So this is the what the transformation by the what that is the induced competence with the help of the chloride treatment. We are giving the calcium chloride treatment, and first of all they are subjected to the cold treatment at 4 degrees centigrade, and after that suddenly at the temperature is raised to the what that is a 42 degrees centigrade for short time or the for 2 minutes. It makes the what this membrane permeable to the what that is a plasmid, and so that now the plasmid will enter within the what that is a this cell. So this plasmid having the what our desired gene, and so the such type of the cell which having our desired gene with the plasmid, this we called it as a what the transformed cell. That process is called as a transformation. Then the next is the what the transformation by induce. Next is the what that is a phage infection, or that is if the what the DNA molecule is introduced into the host cell by action of the what that is a by using the instead of bacterium, if you are using the what that is a the phage molecule or the bacteriophage, then the process is called as a transduction. Transduction is a process of transfer of bacterial gene into the host bacterium with the help of phages. And even in the recombinant DNA technology, we use the word that is a specialized transduction process, and that carries our word that is a the desired gene into the word that is a the bacterium. So these are the what the two processes or important processes which are utilized for the what that is the introduction of the DNA into the host cell. What are these? One is the what the transformation by the heat shock, that is means first it is kept at the 4 degree centigrade and the calcium action of the calcium chloride, and suddenly the heat shock for the what that is a short period of 2 minutes at 40 degree centigrade, and the cell will get transformed. And the next process that is called as a what the calcium chloride induced transformation or induced competence, and second is the what that is a transduction. So this is regarding about the ligation and the what that is a how you can transfer the what that is a bacteria recombinant DNA molecule into the host cell. Thank you.